Om Swastiastu. We are small group discussion B8 from Udayana University School of Medicine. We would like to present our student project about Fasciola Hepatica. These are our group members. This is the overview of our presentation. This presentation has three major parts: introduction, literature review, and conclusion.
My name is Doi Putri Sakarajaya Kanini and I will explain the background of Fasciola hepatica. Fasciolasis is a zoonotic parasitic disease caused by the trematode worm genus Fasciola species Fasciola hepatica. In humans and ruminant livestock, it has been identified as a re-emerging disease. The World Health Organization estimates that 2.4 million people are infected with Fasciola species and 180 people are at high risk of infection in more than 60 countries. In Indonesia itself, the prevalence of fasciolasis varies from 60 to 90% depending on the season and the region. The livestock sector is one of the essential aspects in Indonesia's economy. Therefore, the re-emergence of fasciolasis can hugely impact our economic condition and need to be learned more.
Thank you for the opportunity. My name is Rose Velani and now I will tell about the problem formulation of this student project. First one is, how is the epidemiology of Fasciola hepatica? What is Fasciola hepatica? What is the morphology of Fasciola hepatica? How is the life cycle of Fasciola hepatica? What are the symptoms of fasciolasis? What is the diagnosis of fasciolasis? And lastly, how to prevent and treat fasciolasis. The objective of this student project is to describe the epidemiology of Fasciola hepatica, to describe Fasciola hepatica, to describe Fasciola hepatica morphology, to describe the life cycle of Fasciola hepatica, and to describe the symptoms, diagnosis, prevention, and treatments of fasciolasis. This paper aims to understand Fasciola hepatica, which includes the epidemiology, definition, morphology, and life cycle of Fasciola hepatica. Moreover, we also covered clinical symptoms, diagnosis, prevention, and treatment of fasciolasis.
Thank you for the opportunity. My name is Diana Ningrum. We're going to chapter 2 and we'll start with the epidemiology of Fasciola hepatica. So, Fasciola hepatica is one of the causes of fasciolosis which occurs mainly in areas with a temperature climate and rainfall areas in the world, some of which occurs in tropical climates. The parasite of Fasciola hepatica resides in the liver and biliary ducts of a wide variety of hosts that is usually ruminant animals, such as cattle, rats, and horses. Fasciola hepatica can infect the host and continue the life cycle within the host body. Humans can generally infected by this parasite through diet or eating habits in population or individuals. This can occur in many parts of the world, especially in areas where people consume freshwater plant and drink water contaminated with metacercary that spread by a vector of gastropod snails from the family Limnaceae. The disease that caused by Fasciola hepatica has become a health problem around the world, with an estimate 17 million infected. In Indonesia, previous studies have stated that the parasitic infection is between 60 to 90% of the cattle and buffalo as dominant ruminant in Indonesia.
Thank you for the opportunity. My name is Igderian Nananarnatan, and I will explain about Fasciola hepatica. Fasciola hepatica is a parasite, also known as fluke or flatworm, from the class Trematoda phylum Platyhelminths. The trematode Fasciola hepatica, or also called as sheep liver fluke, causes a disease called fasciolasis. It is infecting the liver of mammals, including human, and it is transmitted by sheep and cattle to humans all over the world. Fasciola hepatica is mainly found in regions with a temperate climate, and it is transmitted to the definitive host, in this case cattle and sheep, by snails that act as effectors from the family Limnaceae.
My name is Tiffany Cecilia and now I will explain about the morphology of Fasciola hepatica. First is adult worm. The shape of the adult worm is large. Their leaves have fleshy flax. Their color is gray or brown. They have a conical projection which contain an oral sucker at the anterior end and a rounded posterior end. It lives in the biliary tract of the definitive host for many years. Fasciola hepatica is hermaphrodite, with both male and female reproductive organs. The second is egg. The eggs are large, ovoid, operculated, bell-stained. The eggs contain an immature larva, which is called the miracidium. The eggs cannot be differentiated and unembryoted when they are just released. The last is larva. The infective form for man and the other definitive host is metacercaria larva.
Thank you for the opportunity. My name is Natania Catherine Suteja and I will explain about the life cycle of Fasciola hepatica. Fasciola hepatica eggs are released to the environment from the host species. In the water environment, these eggs will develop into embryonated egg that can contain the larval phase of Fasciola hepatica that is known as miracidium. After 9 to 15 days, the egg will hatch and the miracidium will search for an intermediate host which is a snail. Inside the snail body, miracidium will form a sporocyst that can contain 4 or more radia. Radia contains a bundle of cerciaria with flagella that can make the cerciaria to be mobile. It will leave the snail body and search for the second intermediate host, which is usually fish and water plants. The cerciaria will stick into one surface and lose its tail, becoming metacercaria, the infective stage of Fasciola hepatica. Inside the digestive system, metacercaria will reach the liver, and in 8 weeks, Fasciola hepatica has spread and reproduced in the human body, thus laying its egg and releasing it into human feces.
Thank you for the opportunity. My name is Raymond Albert Budianto, and now I will explain about the clinical symptoms of fasciolitis. Fasciolitis is an infection caused by Fasciola hepatica and Fasciola gigantica. Humans get infected by fasciolitis by ingesting raw plants infected with metacercariae. Some symptoms of this infection are abdominal pain, fever, fatigue, appetite loss, arthralgia, weight loss, and myalgia. A patient's diet, eosinophilia, and significantly elevated IgE levels can also be important predictors for physicians to suspect if someone gets infected by fasciolitis. On the other hand, fasciolitis may also present without any specific symptoms. Parasitic infections such as fasciolitis can cause eosinophilia as well as induce an eosinophilic leukemia reaction. The normal average eosinophil count in peripheral blood is 500 cells per microliter. Eosinophilia can be classified as mild, about 500 until 1,500 cells per microliter, moderate, 1,500 until 5,000 cells per microliter, and severe, more than 5,000 cells per microliter. If the white blood cell count is more than 30,000 per millimeter cubic, and the eosinophil count exceeds 30%, a eosinophilic leukemia reaction can occur.
My name is Natania Faustin, and I will explain about few methods to diagnose fasciolitis. The standardized method to examine fasciolitis is stool microscopy. It detects Fasciola hepatica's eggs in its chronic or adult phase of infection. Fasciolitis will also be examined by serology diagnostic. This testing will be useful in the acute phase of infection since the specific antibodies to Fasciola hepatica become observable 2 to 4 weeks after infection acquisition. Serological tests to diagnose fasciolitis include immunofluorescence, ELISA, immunoelectrophoresis, and complement fixation.
My name is Sabrina and I will continue the explanation about the diagnosis of fasciolitis. Another method that is considered to be useful to diagnose the fasciolitis is blood test. It includes routine blood work and the detection of antibodies and immune response to the parasite. Imaging tests could also be helpful in detecting of the fasciolitis. It includes ultrasound scan, CT scan, ERCP, and percutaneous scan.
My name is Yevgenia Rindi and I will explain the prevention of fasciolitis. Prevention of human infection is achieved by strict control of the human infection sources in each place, mainly with relation to watercress and other metacercariae carrying aquatic plants, especially in high human endemic areas. Drinking of natural fresh water should be avoided, and the likelihood of human infection in urban areas should even be taken under consideration. And the danger of acquiring fasciolitis by imported foods or chewing plants as cat from endemic countries should be emphasized. So, limitation on consumption of aquatic vegetation, factor control by chemical treatment of water, avoid drinking natural fresh water, and washing fresh produce such as vegetables and fruits before consuming them are the ways to prevent fasciolitis.
My name is Kadek Pramudia Agastecana, and now I will continue the presentation with treatment for fasciolitis. Many drugs are worn to treat human fasciolitis. The first one is amethine, or the better tolerated dihydroamethine, were used widely within the past and still be used today. The second one is butyronol. Butyronol was the following drugs of choice. Occasionally, the patients required a second course to get an entire cure. The side effects including diarrhea, anorexia, nausea, vomiting, pruritus, urticaria, and abdominal pain. The third one is tricarbendazole. Tricarbendazole is the drug of choice at the moment. However, tricarbendazole resistance has been described in many countries in the world. Last but not least is nitazoxanide. Nitazoxanide may be a purified ferredoxin oxyreductase inhibitor with reported efficacy on a broad parasitological spectrum like intestinal protozoans and helminths.
My name is Nanditanas, and now I will explain about the conclusion of our student project. Fasciola hepatica causes a human infection that has become global disease in many countries with an estimated 17 million infected. Fasciola hepatica is a parasite that causes a disease called fasciolasis. It infects the liver of mammals, including humans. The adult form of the Fasciola hepatica is large, with a length of 30 mm and a width of 15 mm. The eggs of this parasite contain an immature larva called miracidium. The diagnostic stage of Fasciola hepatica is when the eggs are released into the environment from the host duodenum via the feces. The infective stage of this parasite is when metacarcariae on vegetation ingested by the definitive host. Some symptoms of infection are abdominal pain, fever, fatigue, loss of appetite, arthralgia, weight loss, and myalgia. The diagnosis of the disease can be done by microscopy examination, serodiagnosis, blood tests, and imaging tests. Fasciolasis can still be prevented by strict control of the human infection sources in each place, and on the other hand, fasciolasis can also be treated with several drugs such as triclobendazole and nitazoxanide.
Hello, my name is Deoputu Adeno Pratnyana and we'll continue the suggestion. So there are 3 suggestions in this student project. The first one is the research and findings on Fasciola hepatica are still developing worldwide. The second one is fewer research, depth analysis, and the discovery of new drugs on how to prevent this bacteria and how to face it when it comes to infections are needed. And the last one is resistance of Fasciola hepatica is also important to be faced so we can reach a better treatment in the future. Thank you so much.
Lastly, these are some references that we used in the process of making our student project about Fasciola hepatica. Now we are at the end of our presentation. Thank you for your attention, and we would like to apologize if there are some mistakes that we made intentionally or unintentionally. We will close this presentation with Parama Santi. Om Santi 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 Om.